So good morning, and again, thank you for joining us for the ARM 2017 State of the Industry briefing. I'm Sarah Haecker Meeks, and I am moderating our next panel, The Gene Therapy Pipeline, Will Clinical and Regulatory Progress Match Patient and Investor Expectations? And before I engage this excellent panel, when I was asked to do this, I was thinking about where I started in gene therapy. And if you can give me 2 minutes, I'll describe sort of a funny story that where I started. And so 22 years ago this month, I started a postdoctoral fellowship in the Institute for Human Gene Therapy at the University of Pennsylvania. My project was to develop muscle-directed gene therapy in the lab, and the first project was to develop a vector carrying full-length dystrophin for treatment of Duchenne muscular dystrophy. So remember, this was 22 years ago, and think about what I had available to myself in the lab at that point. I set out to do it, and in 1995, I cloned the full-length cDNA for dystrophin, which is 14.2 kb long, behind the CMV promoter, because that's what was available to me, and in a bare bones or gutted adenovirus is what we called it back then. And although I had, manufacturing options were very limited, I was able to grow up enough vector to inject a dozen or so MDX mouse animals, the model for Duchenne muscular dystrophy. And in these experiments, we were able to detect functional dystrophin in the skeletal muscle of these animals, but we saw no functional correction. The expression was localized to the point of injection in the skeletal muscle, and it lasted just a couple of weeks. And nevertheless, we presented and published this work and called it an early success. That was 22 years ago. And Dr. Preddy reported this morning, and I'm just going to point out a couple of the facts. There's a couple of the numbers that we presented from the Alliance here. There are approximately 100 companies in this sector we're calling gene modification, gene therapy, in vivo, ex vivo, and genome editing. Approximately 100 companies, both therapeutic as well as enabling technology companies, supporting this sector. Over the last 2 years, I realize this past year was quote-unquote a down year, but there was nearly $10 billion put into this sector alone. We had an approval last year of Strimvelis in Europe, and we are expecting 3 BLA submissions this year, along with a number of very important data events. We're focused on regulatory, we're modeling reimbursement, and we're thinking about patients across a number of indications. So, again, when I think about it, we've come an enormously long way, and, it's meaningful. And so with that, I'm going to move over to my panel, and my terrific panel, and I'm going to have them, to kick this off, introduce themselves, introduce their company, the platform, their approach, and then take 5 or so minutes to comment on the statement that Bob Preddy made this morning is, are we at an inflection point in this industry? And if so, why? What are the indicators? And if not, or what's left to do here? So with that, I'm going to hand it over to Ness Bermingham.
Thank you. 22 years ago, I was just thinking about that. I finished my PhD over 22 years ago, and today, which took me 3 years, and today you can do my PhD in less than a week. So it's amazing how technology has advanced. It's kind of a humbling thing, right? And you talk to some of the scientists and the technologies and tools we use then, they've never even heard of. So Nessan Bermingham, CEO and founder of Intellia Therapeutics. Intellia is a company based in Cambridge, Massachusetts. It's focused on CRISPR-Cas9 for gene editing. The company was founded out just over 2 years ago, and really it was around the invention or discovery of an endogenous system in bacteria and archaea to allow you to cut DNA. So when you hear about conversations about molecular scissors, it's a pair of molecular scissors that allows you to go in and cut in a very targeted fashion the DNA within your cell. Now, when you think about that, well, why is that important? Well, within every single one of our cells today, there's a repair mechanism. So if I take one of your cells out of your body right now and look at it, there's anywhere from thousands to tens of thousands of cuts within your DNA. And there's a repair mechanism in there to actually allow it to repair it. So by us going in and actually cutting a DNA in a very targeted fashion, we can then go and do various things by driving your repair mechanism to repair that site of DNA cut. So for example, we can insert a fragment of DNA, we can switch off a gene, or we can actually repair a mutation or a genetic defect that's present within there. So within Intellia Therapeutics, what we're doing is we're taking the technology and we're taking other technologies in combination with it to allow us to go in and start to explore and make modifications in a cell. Both cells that we've taken outside of your body through partnership that we have with Novartis, and cells within your body through partnership we have with Regeneron, where we deliver the drug CRISPR-Cas9 directly into the body itself. So to put this in perspective, what does this mean potentially as we think about the future? Potentially in the future, you can walk into a physician's office with a genetic-based disease. A physician will send you to be sequenced or send your DNA to be sequenced. The mutation within your DNA will be identified, or the modification within your DNA that's driving this identified. And with a single or a handful of treatments, you are cured. When we look at data that we've presented so far around this, single injection into an animal, modifying over 60% of the cells within the liver, and reducing circulating protein within that animal that's toxic by over 80%. So I think as we think about these technologies, we're in a very strong position to move forward. We have two partnerships, as I mentioned, one with Novartis, predominantly ex vivo in the area of CAR-Ts. We also have a subdivision called Intellia to expand the ex vivo application of the technology. And then in vivo, we have a partnership with Regeneron Pharmaceuticals around predominantly the liver, but also exploring other applications outside. Delivery is a primary aspect as we think about these sort of therapeutics or technologies. We're using a delivery system called lipid nanoparticles as our first delivery vehicle to allow us to actually target into the liver. So to answer the question, when we think about where we are today, we've been on the investment side. My history is venture capital predominantly, and also working on Wall Street as a research analyst. When I started on Wall Street, I was working with companies such as Onyx with 015, Introgen, Genvac, Diacrin, Targeted Genetics, many of these companies that are no longer around today, or they've changed their business strategy overall. As we think about the challenges that they faced, they were multiform, either delivering foreign DNA or foreign nucleic acid into the cell. People really didn't know what a cell did under that structure, right? When you think about assays for releases, when you think about manufacturing and scale up, when you think about overall response or activity of a gene within a cell that you're putting in there, what does it actually do? Never mind bioinformatics, never mind understanding genomics and DNA. If you think about when the human genome was sequenced, 3 billion approximately nucleic acids being sequenced in there, understanding how do they relate to disease and how do they drive it. So fast forward 20 years from when that was started to where we are today. When we think about the information that's been generated over the last 20 years from work, as you think about Duchenne muscular dystrophy as an example, start thinking about lentiviral delivery systems, adenovirus, AAV, lipid nanoparticles, conjugates. As we start thinking about modification of nucleic acids so it's stabilized within cells. So as we think about 2'-O-methyl modifications, for example. As we think about our genomics and what we've learned from genomics, and also as we think about cells, taking cells out of your body, modifying them, keeping the cell as it needs to be, and keeping it robust before putting it back in. And then stuff that we've learned in the immuno-oncology space. There's been a tremendous amount of data now that's come together across multifacets as we think about biotech. That's actually really allowing us to move forward extremely rapidly. BioMarin announced data today. We've got great data that Bluebird has been announcing. Avexis, as we think about SMA, has been announcing, and clearly Spark announced more data, I think yesterday or this morning. So when we think about where that's brought us, we're at a very real inflection point as we think about moving forward to the future of these alternative or new therapeutic modalities to address and potentially cure genetic-based diseases. Will it match investor expectations? I personally believe it will. As historically an investor, I believe that it will. From regulatory progress, though, that's one place that we need to spend a lot more time, just as we think about that regulatory path forward to allow us to become more flexible in moving these programs towards the clinic, into the clinic, and getting approval on a more personalized basis as we think about some of these applications. And then reimbursement, as the last panel talked about, that's absolutely going to be a critical aspect of it. But people haven't had to address these issues before because the technology wasn't quite there or ready for addressing those elements of it. So with that, Andrew, Matt.
Good morning, everyone. My name is Matt Patterson. I'm the president and CEO of Audentes Therapeutics. But before I tell you about the company, I have to, listening to Ness talk reminds me of a funny story from the ASGCT meeting a couple of years ago that I went to, actually maybe 3 years ago now. And I was relatively new to the field. I was excited to be at the conference. And I found myself talking to a young scientist. And he said, "Oh, what is Audentes? What do you do?" And I was so fired up to tell him. I thought we had the coolest company and we're doing the neatest things. And I started telling him about the company, and I'm looking at his face, and I'm thinking to myself, why is this guy not reacting more excitedly? He sort of seemed a little bored. He's almost sort of drifting off. I'm fired up. I'm trying to tell the story. What's wrong with this guy? And I finished my story, and he looks at me. He's like, "Oh, so you're doing old gene therapy." I was inclined to punch him in the face, but I instead went and got a coffee. Anyway, he was working on CRISPR-Cas9, wherever he was. But in any case, so we work on old gene therapy. Specifically, we develop products for serious life-threatening rare diseases using AAV gene therapy technology, gene replacement therapy, if you will, to contrast with some of the gene editing work that Nessan described. We're based here in San Francisco. We have 100 employees based here in the city and in South San Francisco, where we also have a GMP manufacturing facility that we've invested in, which is a subject we can talk about. We have 4 programs in development. They all have those same hallmarks of being serious and life-threatening, monogenic in nature, well-understood biology, and for those reasons we feel are a great fit for AAV science as we apply it today. And we're very thrilled to be moving into the clinic in our first 3 programs this year and expect to receive preliminary data from all 3 by the end of the year. So a really exciting time for our company specifically, but also certainly the field. And Ness just mentioned a couple of the examples of why I'm personally really excited. I mean, every year it's been amazing progress. 2016 was even more. And for me, it was highlighted primarily by clinical data. And primarily, not just in the extension of some of the ophthalmology knowledge that we had in Sparks data was encouraging this morning, but really the news in hemophilia A and B from multiple companies, BioMarin's data yesterday, another highlight, and Avex's data in SMA, which was particularly relevant for us because 2 of our programs are neuromuscular diseases. We use both AAV8 and AAV9, but just like they're working in SMA, we apply a systemic administration to hopefully achieve benefit in what are really terrible diseases with no available treatment. So those examples were remarkable to me and an incredible step forward. You combine that with the continued obvious enthusiasm from an investor standpoint, I think we are at an inflection point for sure. That being said, I do think it's important that we're not just backslapping and high-fiving the whole time. There's a lot to be learned even in the gene replacement therapy community. And that's going to come with more data. There remain a lot of theories about how the science will work in the long term, how durable these responses will be, the importance of various immunological reactions that you see upon administration, but only with data will we truly be able to answer those things. So I look forward to more data to help us all learn and apply that as we go forward. But moving forward, there's a significant amount of work to be done. It's great that there's so many programs going into the clinic. It's great that so many are approaching commercialization. But we have a lot to prove still in the regulatory operations, I'll call it, mostly manufacturing related, and commercial, primarily around the reimbursement model, I suppose, but also just execution. It's such a novel approach to providing a drug to a patient, for example, or a treatment to a patient. We're breaking a lot of molds with what we do, and so there's a lot to be proven. And I think when we can get there, you'll bring even more and more investors into the field and confident, and we can really be rolling with success on the commercial side from a variety of companies that are on the cusp of that now. So with that, I'll stop.
Thank you. Chris Mason, Chief Science Officer at Avro Bio. Ness, I think 22 years ago, we were in the same lab at Imperial College, I have a feeling, and we were really doing old gene therapy. We were trying to cure cystic fibrosis with liposomes and mini chromosomes to cure cystic fibrosis, and we had all sorts of wacky ideas. the world has moved on. I'll come back to that, but I think that's the reason we're now getting towards an inflection point. Avro Bio is a new company started in 2015 by Jeff Mackay, who is known to pretty much everybody in this room, who was the former CEO of Organogenesis and chairman of ARM. Kim Warren, who was head of process development for Lonza for many years, and myself. And we have a ex vivo gene therapy company using lentiviral vector for 2 platform technologies which we licensed out of academia. The first is an oncology platform where we modify the patient's own tumor cells so that they secrete IL-12, and that delivers targeted IL-12 to the patient's immune system to trigger a cytotoxic T lymphocyte response. Our other platform is our rare disease platform, where we take hematopoietic stem cells from the patient, modify them again with a lentiviral vector, and put them back to replace a missing enzyme. Our first 2 indications are AML for the oncology platform and Fabry disease for the rare disease. And both those platform technologies are now entering the clinic and will be in patients very shortly. So to answer Sarah's question, I'm not sure about the inflection point. I think it's more of a tipping point. I think what we saw was 20 years ago, we saw a lot of technologies trying to come forward, and they all look very promising, and you can get a lot of them to work very nicely in the mouse, as I think we saw many times. And those technologies have matured over the past 20, 25 years, mainly in the academic setting. There really was a sort of nuclear winter for gene therapy in the 2000s. And it's really only as the academics got the science to really work and start to perform. And as we heard earlier from Bob, a great many things now are progressing well in the clinic. They're in phase 2, maybe pivotal, phase 3. But we are still stuck as to what the infrastructure is that's actually going to bring those forward. So the science is moving forward extremely well, and the clinical data is moving forward very well. And I agree totally with Matt that more data drives the whole thing, but that data is beginning to appear. I think where the sticking points are, and I think these 2 are improving, but not improving as fast as we're managing to move these forward in the clinic, are things like the manufacturing. It still takes a great many months to manufacture, for example, a lentiviral product. It will take maybe 3 to 4 months just to make the plasmid as the starting material for the viral vector, another 3 to 4 months to make the vector, and then another few months to actually make the cells themselves with the genetic modification. So you're talking about a process that the very quickest you can do is way over a year. These are long timescales when you want to plan them into your pipeline. So I think the manufacturing, we are seeing a lot of people trying to do it in-house. We're seeing an increasing number of contract manufacturers. We need a lot more joined up in that respect. We need a lot of standardization, which we don't have. I think other areas are things like regulation, trying to get some sort of harmonization. Europe has been mentioned earlier as pretty fragmented, and yet certain countries in Europe are very favorable towards gene therapy because their knowledge goes back 20, 25 years. They did the boy in the bubble stuff in the 1990s, some of which unfortunately got leukemia, but that gave the Europeans a lot of experience. So countries like France, Italy, UK are very good countries to go to if you want to do a gene therapy product. I think other areas which we aren't really fully au fait with is really the people. We need more good people. I mean, academia has produced a lot, but they're not the industry folk, not to give us the industrialization and the scale up. And I think there is real pressure now to actually have more good quality people available. And finally, I would say that Big Pharma are taking a very active role in trying to actually develop some of the technology. So if we look at people like Pfizer, who have got a very strong AAV platform technology, and also GSK with their lentiviral one, these are involved hundreds of people who are doing some very high quality process development and other research work.
Thank you, Chris. So I'm going to run with the manufacturing one for a minute here. There's a few topics that we wanted to cover that we all talked about prior last week, but I think that if we can run with manufacturing a little bit here, each of you with different perspectives, and really thinking about what you said, Chris, is, in process development is, there's an engineering component needed here. That's a different skill set that we've traditionally not had in the old gene therapy labs, right? Can you each comment on how you're approaching manufacturing and what you think the immediate hurdles are, and how do we get to commercial scale manufacturing?
Okay. For us, one of the reasons we started with lipid nanoparticles, not only because of the size and everything else, these are chemically synthesized structures. So, from a manufacturing standpoint, as you think about moving outside of the ultra-orphan or orphan indications to treat much larger patient populations, one of our indications we're looking at, for example, is HPV, right? You need to be able to scale up at not only from scale-up size, but from a cost standpoint to enable you to do it. The other elements that are present within the drug itself is the nucleic acid. In our case, it's RNA, right? So how do you think about scaling up that manufacturing? That's something that we're working on. I look back, though, at history and think about, for example, biologics, right? If we think about antibodies, if any of you remember back, 10, 15 years ago, there was a big discussion and a big issue that we had in our industry around the actual scale of manufacturing, right? And the issues that people were faced with as we thought about rolling bottle manufacturing, et cetera, that was present within there. Now, fast forward, because we had so many antibodies starting to come through, there were significant changes that were driven on the actual manufacturing process that allowed us to scale up. So we were able to move from, not only from a cost of goods standpoint, but an overall global supply, where today people in their kitchen on a weekly basis can dose themselves with Humira, right? So I think that there are some very significant manufacturing issues that we face, that we, from a scale-up moving outside of the orphan indications, one of the reasons why a lot of companies focus on orphan is because that's where you can manufacture for. But we're starting to address it. It'll take us a little while, but we certainly will. I believe we will certainly get there.
So it's been quite an experience for me getting to know gene therapy manufacturing, AAV manufacturing. I would say from the start, when I started the company end of 2012, I did not appreciate the complexity of the science and the challenge ahead for manufacturing these products. I'd been involved in biologics product manufacturing for many years, but quickly came to realize that the state of the union for at least AAV manufacturing looked a lot like, late 80s, 1990 CHO cell protein manufacturing, in that there wasn't much external expertise. Really, the people who knew how to make the products at a technical level were academic experts who'd worked at small scale and adherent 293 cells, and that there wasn't a lot of knowledge externally that you could turn to, both for the science of how to manufacture the product or the capacity needed. Now, what problem you're trying to solve, it varies. If you're in ophthalmology, you need far less material. We're in liver and muscle. And in muscle in particular, all the data begin to tell us across the SMA program, our preclinical data in some Duchenne programs, that you need around 10 to the 14th vector genomes per kilogram per patient. And even in children, that's a lot of material. So the challenge was real for us. And so when we went looking at options, we were unsatisfied, frankly, with what external options were available. And we went through an exercise internally. We said, what would it take to renovate an existing warehouse or some other space nearby in South San Francisco to achieve our needs for the coming years? And how long would it take? What would it cost? And we got fortunate through that exercise, actually, in that they came back with an initial estimate. It wasn't as terrifying as I thought it would be, but it was a little scary given that we were preclinical stage, right? But we also found an interesting opportunity of an old building, been used for biologics manufacturing for 20 years. It needed a lot of TLC, but for a very relatively modest capital investment in a shorter period of time, we took on that challenge. And we renovated that, and we now have two 500-liter bioreactors making GMP material for all of our programs. And it's a strategic decision that took some courage, to say the least, with no human data. We're investing in a biologics facility. But every day that goes by, we're thrilled with the choice and have expanded that greatly to include a full research effort that involves molecular biology, analytical method development, process development for the small scale to enable the large scale. So it's an incredibly complex field, but one that we've at least decided to invest in internally. Of course, there's a ton of work going on at CMOs and elsewhere to ramp up rapidly, but it's still challenging. So we're thrilled with the decision. I do think it's going to do a lot for us, improve the chances we hit our timelines, keep our costs down. Interestingly, I think protect know-how. We're all going to learn how to do this at a larger scale over time, and there's going to be a lot of lessons learned, and so having control over that excites me. also control over the analytical side of this work. It's not just the production, but the analytical tools needed. This gets into the regulatory environment too, but having the analytical tools in-house to characterize how we make these products is going to be critical. In particular, when you realize that we're going to go through the regulatory challenges over time of scale up and needing to have dialogue with the authorities. And so if you have the tools in hand to help clarify that your product is comparable, you'll be in good shape. So we followed a fairly unique path, I know, for companies in our space, but hopefully that gives you a sense for the challenge.
Yeah, I think we're quite different in that respect, in that Matt and Ness both are very much allogeneic approaches. They can make a large batch up and treat an infinite number of patients. The approach that we're using with lentiviral gene therapy, same as with Bluebird, is that these are autologous gene therapy products. So we have a much greater complexity of manufacturing. We're actually going through the CMO route. That's our chosen route forward. But I think probably the one thing that makes the biggest improvement is what was mentioned earlier, going from needle to needle as a closed system. And I think that the closed system is really, we're certainly going that route. And I think we could do with more manufacturers producing turnkey closed systems. I mean, at the moment, the Miltenyi one is probably the preferred choice for most people. But I think there is an opportunity there for bioreactor manufacturers to start to produce more closed systems. They need better analytical devices with them, better control, better monitoring. But I think that these are the things that are going to make a really big difference to our sector. And then I think once you have those very tightly controlled closed systems, you can then, as was also said earlier, produce the stuff in a much more lower classification of clean room, which again then starts to bring the cost down and make the manufacturing price more palatable.
I'll just add one quick thing, Sarah, that we don't want to lose sight of. It's not just the operational challenge of making enough material. I think it's also making the right material. With time, I suspect we're going to learn more and more that the specific design of the expression construct and your control over its manufacture, your testing of it to confirm that will inherently dictate the long-term efficacy of the product because there's growing evidence that those elements will play a role in the immunogenicity of these products and therefore their long-term benefits. So the manufacturing is both an operational challenge but also the quality systems, the nature of the product you design, and how you make it on a regular basis, how you test it on a regular basis, I think will be critical to the clinical data and eventual regulatory approvals and commercial success that we all hope for.
Good. Okay, I'm gonna switch gears and just touch on this one very, very quickly, and then move to regulatory and market access. But just very quickly, there have been a number of key improvements in technology and science over, let's say, we're 22 years, right? 25 years. There's Matt's old gene therapy, but there are next-gen constructs within that old gene therapy, including, novel capsids, novel promoter systems, optimizing these expression cassettes. Can you just comment briefly on any of these technologies that you think are critically important to, and could they accelerate pipeline development? Maybe Chris, you want to start?
You're absolutely right. The world of the vectors is going to change very rapidly. There's some beautiful work going through the academic world at the moment, changing the envelope so you can maybe target cells specifically using these gene therapies directly in vivo. From a lenti perspective, the one big area is the ability to make stable cell lines. At the moment, to make a lentiviral vector, we use 3 or 4 plasmids, which we put into a 293 cell. It produces the vector for a short while, but the vector actually kills the cell. And so we have to do it all over again. It's a transient expression system. So it means the batches are very expensive, and they're also highly variable. And there's a lot of work going on to try and see if you can make permanent cell lines that will continually spit out a lentiviral vector. I think once that is done and readily available, then we'll see the cost come down and we'll see lentiviral gene therapy much more amenable. So stable cell lines.
Yeah, stable. Matt?
Sure. So, we leveraged what we believe are some of the optimal science vector, AAV vectors and knowledge going forward to hopefully achieve near-term success. So what do I mean by that? We use vectors that are pretty well understood today. We license our vectors from Regenerative. We use AAV8 and AAV9. We have experience with those vectors in humans, 9 now with the SMA data, which is encouraging. But these are well characterized. There's a lot of talk about improving on the vectors for the future, and that's very interesting to us. But in the near term for me, it's really about driving to clinical data and hopefully commercial success. So we're starting with AAV8 and AAV9 vectors known from Regenix and the early work at Penn. We certainly take a thoughtful approach to promoter selection and other elements of the expression construct. Our head of R&D, John Gray, has a lot of experience from time in the field in the academic world, who helps us design these constructs in a way that's proprietary, that we hope translates to improved safety and efficacy over time. There's a tremendous amount going on that's interesting to us, whether it's vector science, promoter science. We're more focused on manufacturing science for the near term. I'm interested in getting involved there. I like to joke with my team that I'm not burdened with an advanced scientific degree like so many of them are, and my panel mates, so sometimes I keep it maybe too simple. But hopefully that helps translate to near-term success, which let's be honest, in the very near term, those new developments are really interesting, but in the coming years, the success of the field will largely be driven by more clinical data, which really is applying the science as we have it today and getting to approvals and then continuing to build on that over time.
So certainly I think delivery, there's some advances taking place, not just in viral, as we think about moving outside of viral into more synthetic-based systems. For cellular targeting, I think that's really important as we think about being able to direct it to specific regions of the body and do it in a very targeted fashion. I think there's a lot of stuff we still just don't know. When we think about the complexity of our genome, the complexity of a cell, and then the complexity of our body, what happens within these things. The sensitivity of our assays, we're really starting to get a better sense of that, being able to hone down as we think about the cost of sequencing that allows us to go in and sequence and see exactly what's going on within the genome after we've delivered a viral delivery system in there or delivered a CRISPR-Cas9 system in there. I think that's very important. I also think the bioinformatics genetic disease, understanding the genetic association studies that we're doing, the large population studies that we're doing, to me that's extremely exciting because we're starting to understand that we originally thought there were these massive amounts of genes that were present within our body that was driving all of these things. And what it turned out was actually that's not the case. There's a very limited number of genes that actually drive the proteins in our body. And that what we thought originally was junk DNA that was there just as a remnant from our ancestral experience actually is extremely controllable regulatory sequences. So starting to understand that and how to modify or impact that to allow us to treat disease and address disease, moving from monogenic disorders where a single gene, cystic fibrosis, for example, to a specific disease, to far more complex, Alzheimer's disease, bipolar, schizophrenia, Parkinson's disease. How do we start thinking about treating those? I think we're starting to see a lot more work going on because we have tools to allow us to start to break this apart or interrogate it in a more rigorous fashion. I think that's extremely exciting work going on right now.
Yeah. Good. How much time do we have? 10. Okay, good. So 2 more topics that I wanted to make sure that we covered, and you all have commented on them to start, but regulatory and so commenting on regulatory issues, and I would just say that, and kudos to Michael Werner and his team on the 21st Century Cure Act to get this RMAT designation. I mean, it sounds like we as the gene therapy and genome editing sector here now have our work in front of us to be included in that designation and to do work there. Can you just briefly comment on what you think the major hurdles on the regulatory front are in the next 2 years to 5 years? I mean, what's going to move the ball forward here for our companies? Sorry.
So, first of all, I would say I'm generally encouraged by the regulatory environment and our interactions. They've been more on the IND enabling side, of course, as I mentioned, given our stage of development, but it's been a really receptive audience. We heard that a little bit from the earlier panel, too, that I think the authorities are eager to see this science move forward and see programs begin to gather clinical data. Getting to approval is the big hurdle here. Now we've got a couple in Europe that have been achieved. It's important that we get there in the U.S., and hopefully we'll be sitting here next year talking about an approved AAV product and a couple CAR-T products, perhaps. And so, but getting to that milestone is key for all of us. And so, if I had to think about the areas of most interest to me on the regulatory front, is how can we collaborate with the authorities to get to a place on CMC with regards to defining and validating our manufacturing processes, establishing specifications for proper control that makes sense for the products as we understand them at the time. That, I think, is going to be an interesting conversation that we need to engage in early, so we're trying to do that now as well. And then I think pivotal trial design interests me a lot. I've been really encouraged by what we've seen from some of the news flow out of companies that are advancing into late stage. I guess I would highlight Bluebird and Avexis as far as the interactions they've had and what seemed to me to be a good degree of creativity from the agency in particular about study design, necessity or not of control groups, numbers of patients required. It will be interesting to think about how much data, how long do you need safety data in particular to feel comfortable approving it. But I feel like we're drifting in the right direction there. It definitely requires a lot of engagement, and hopefully companies take that very seriously. We certainly intend to, and I think at least with CBER, they're very interested in being as helpful as possible.
Yeah, I would agree with all those things. I mean, I think that there are still some really big fundamental questions to be resolved. I think things like dose and potency, we're still nowhere near being able to define them tightly enough, or what is an acceptable range. So I think they're dialogues that we're going to need to have with the regulators, and I think the regulators are keen to do that. I mean, I think globally, Japan, Europe, US, we see regulators keen to learn. I think we heard that earlier, and I think that's true. But I think that unless we can start to get our arms around this sort of very fundamental characterization of exactly what our drug product is, it's going to be very challenging for quite a time.
And challenging again, especially in the autologous model too. I mean, you've got a different set of challenges there as well, I would say.
Yeah, but even in the allo, even with things like AAV, having consistent batches is very hard. You can have a very large number of empty capsids, for example. So I think it's right the way across the space. I think we need to. It's very much a work together to get these potency assays and what exactly is a dose. We're not talking about a small molecule drug where you pop it in and then once it goes into the patient, it starts to decrease in numbers. These may expand in numbers. So we have quite a different situation, and I think that's something for the industry to work together to try and define some of these really critical parameters.
And I think the standards coordinating body and the work that ARM has begun with a number of other groups is going to be important, in addressing some of those issues. Please, Ness.
So, I think the regulatory bodies globally, the amount of work and the area that they have to focus on or deal with is tremendous. Even when we just talk about AAV, we're not just talking about one single virus, AAV2, let's say. We're talking about a whole range of AAV viruses that are being manufactured in different ways that they need to deal with. So I think the immediate need is certainly on the resource side, right? As I look at putting more consistent guidelines in place, adaptive studies, as we think about the studies that we're doing, for many of these therapies, there is something that's entering the clinic for the very first time, be it a new promoter, be it a new virus, be it a new protein that's being expressed within it. So I think as we think about how do we adapt our clinical trials to enable us to actually learn as we're dosing a patient or treating a patient and moving forward. I think longer term, I mean, if you look around the room in here, right, every single person in here is different. One of the things that we've done when we think about small molecules, or we think about antibodies or recombinant proteins, is we've made an assumption that we've had to, that you inject that into a patient and you'll see a consistent effect, or that, frankly, within specific races, people are going to be the same. When we start thinking about genetic drugs, though, that's different. That's not the case. Even a promoter that will drive very strong expression in cell type from person A will not necessarily do that in cell type person B. So as we start thinking about a more personalized approach here, I think we've got to start thinking about what are the implications of that. How do we think about data in helping us, in educating us as we think about moving that forward? With all due respect on the gene therapy side right now, one of the areas we've been pushing has been get as much virus kind of in there, right, and hope you can express as much protein as you possibly can, because too much protein hopefully won't do anything detrimental to the cell. And that's certainly not the case. I think we need to be very careful about that. But also when we think about genetic disease, it's not a cystic fibrosis patient is like a cystic fibrosis patient is like a cystic fibrosis patient. You're talking about over 1,000 different mutations within CFTR, right? Delta 508 is the one I think most of us probably have heard of because that's the most frequent one. But you have people out there that have maybe 3-5 people with a single mutation in the whole world. how do we think about treating those people? We can't run a clinical trial for them, but are they a less right to a therapeutic? So as I think about looking forward longer term, 5 to 10 years, it is, are there structures or systems that we can start putting in place where we can actually start to make it truly a more personalized approach, similar to what we do with the clothes that we wear, the shoes that we buy, right?
That intersection of individualized medicine with advanced therapies. So I think we have a couple more minutes and one more topic I wanted to cover, and that is, just how about some comments on the reimbursement, market access, the modeling that's begun in and around these types of products being potentially curative products, needing, having a durable profile. Can you comment on that? I know that there's been some work that's begun from a number of different areas, but it'd be great to get your thoughts on that from the panel.
Okay. So reimbursement is obviously a hot button, right? And there's a lot of considerations going here. There are a lot of models that have been thrown out and discussed around this. Annuity seems to be the one that you tend to percolates up to the top a lot. What people seem to hear in the U.S., there are some places where an annuity model may actually work in the U.S. With the current structure, I think it's going to be very difficult. You have a number of considerations. So the first one is, as you think about payers, the average person stays, about 3 to 5 years with their payer before they move on, right? So from a payer standpoint, a new model doesn't quite work for them, and the transfer over, why will a new payer necessarily pick them up? When we think about a patient themselves going in for an annual checkup, if you've cured a disease, right, the drive to get that patient back into the physician's office is very challenging. They do not necessarily want to be reminded that they actually suffer from this disease or were exposed to this disease early on. So when we start thinking about reimbursement, it becomes extremely challenging. I suspect that we will see systems come in place whereby it's payout for performance over time. If you look at, there may be a bullet payment that takes place up front, and then there are maintenance payments that are paid so long as that patient is still within a system, and there's an automatic handover to a payer that I think we need to start exploring. So I think their model isn't there, and we haven't seen one that will address all the issues yet. People are very clearly trying to work on it. Given the inflection points that we have with Spark, potential approval this year, given what we're seeing in the broader, more active cell therapy space, given the work that Bluebird of Exis are doing, it's going to drive it, right? And with the recent SMA approval, $750 for your loading, and then, $350 or $375 maintenance, that's an interesting level that we'll see how the payers actually respond to that.
Yeah. And I'll just, before the two of you, one other comment that you made last week, someone made last week when we were discussing is, what's the physician's response going to be to that? When do we start working with the docs on this and how they're treating, with potentially curative drugs?
Yeah, that is always a challenge. I think the clinicians are becoming more comfortable. Most are fairly conservative and don't necessarily want to move into technologies which they don't understand. But I think there is a gradual increasing awareness amongst the clinical community of the benefit of these therapies. I think initially they will be in the tertiary centres, and then they'll fan out into not so much big hospitals. I think what we have to do is really start to that dialogue early. And when you're doing a clinical trial, you're getting those clinicians familiar with your therapy, and they're going to be probably the first ones once you have got marketing authorization.
Matt, last word?
Rather than repeat what's been said, obviously it's a critical subject. I would say that no one in this room will be happy if we're lucky enough to develop incredible data from a bunch of these programs and then patients don't get them. It's just not acceptable, right? So we have a lot of work to do, and we do need to take the commercial issues very seriously, along with the development and scientific challenges that we all face. So there's things individual companies can do to build development plans that make good sense, that set you up for future success. For example, we're trying to be very thoughtful about what data we're collecting during clinical development to evaluate the burden of the disease in each patient, for example, with these terrible diseases we work on. We try to engage as much as possible. We've engaged already with NICE before we were even in the clinic. We had a conversation and got some feedback from NICE about our development plans to try to do it as thoughtfully as possible. So there's things you can do on an individual company basis that we can all do that are, I think, important to future success. And then there's things broadly we should be doing as an industry, and it's a plug for ARM, but it's important. ARM is very focused on this. Maury mentioned in the beginning, it's a strategic priority, ensuring that we're working all together as a field to engage in all the different stakeholders to come up with a system, in particular in the U.S. I mean, the U.S. has the bigger problem in that our system currently is just not well designed for an annuity type model, and that can be defined in multiple ways, but an annuity type model, which is probably the long-term answer, right, for a good balance of proper reimbursement for companies who've invested so much time and money, the value you're bringing for these patients, but also with an eye towards coming to market with a limited amount of data when you really want to be reimbursed for maybe a lifetime or decades of benefit. So, and then in the short term, there may be patches that don't solve the problem right away. And finally, there won't be one size fits all. We can certainly say that. We're not going to ARM or anyone else shouldn't be advocating for this is the system everyone should use. But let's make sure that there are a range of options, especially here in the U.S. where we have this challenge structurally, to ensure that every product that gets developed can get to patients. Some of them are going to be one-time payment. That's fine. Some will be more over time, and that's great too. It just depends on each individual case. So, but let's take the work seriously.
Thank you. We're to the end of this panel. Will you please help me in thanking our panel?